I should start by saying that I was one of a group of immigrants that came into the field in the 1980s with new techniques. And that was a time where people were applying new techniques to the study of rhythms and finding out things they couldn't find before. I had been working in invertebrate neurobiology and looking at an oscillatory network that drove the protraction and retraction of the mouth of the snail when it was eating. And when we moved to the University of Illinois from the University of California at Santa Cruz, I collaborated with my husband for about a year, and then I decided that the University of Illinois would never need two people working on the same molluscan neuron. And I was looking for a project that involved cell signaling because that was a new area then, and I was very, very interested in second messengers and how they could cause long-term state changes in cells. And as a class project, Dan Green, working with Raynard Gillette, my husband, decided to apply the method that had been used in the hippocampus to study long-term changes, which is well known as long-term potentiation. They decided to apply it to the hypothalamus and see if you could get a sustained rhythm or some manifestation of timekeeping out of this piece of the brain. And it really sounded like the strangest thing in the world to do, that you could take a piece of brain out, put it in a life support system, and ask it to keep time. However, this is what was done in molluscan systems, where you weren't asking them to keep time, you were taking the brain out and recording from all the roots and saying, can I get something that looks like walking or running or swimming by measuring the roots? In other words, fictive, so-called fictive behavior. So Dan took the slice of brain out in this class, graduate student class, and recorded for 24 hours, and he found that neural activity, if you just sampled basically at random throughout the nucleus, went up in the daytime and went down at night. And if you took an animal from a reverse lighting schedule and did the same experiment, put it in the same dish, it also went up during its day and down at night, but that was inverted from the first sample, so you had two rhythms that were out of phase and went like that. And what a great finding for a classroom study. And I remember Dan saying, you know, this is so complicated. My wife hates it when I sit up all night, and I really would like to look at membrane ion channels. And I thought to myself, you know, I've always liked complicated things. It's like a personality flaw. So why don't I look at the brain slice? And the first thing we did was try to find out if you could adjust the phase like an animal does when it's in a different lighting cycle. And I tried applying an analog of the second messenger cyclic AMP because it is the most widely known second messenger system. It's most ubiquitous second messenger system in all living things. And it's what I'd been working with in the molluscan feeding network to change its rhythm. So I applied this analog, and lo and behold, the phase changed. In fact, we applied it in the middle of the day. The next day was advanced. And we took this work to the Society for Neuroscience meeting, showed it, and Rebecca Prosser was working with me by that time, and I remember showing the results there, and everybody said, well, gee, Martha, I admit that the peak is moved, but you haven't gone to the second day. You know, maybe it's just something that was established temporarily. It's a transient. So you've got to go to the second day. And that was actually quite a confrontational meeting where the chair of the session didn't let anybody else ask questions and really hammered us on what kind of progress were we making with this if we couldn't go another day. So we went back. I said, Rebecca, we got to go to 2 days. And so we tried all these manipulations to get the slice to live for beyond, because we were minimal salts and glucose, and that's a very, very defined, but there aren't any growth factors or anything that might sustain long-term life outside of the brain. And the more we did, the more our alterations look like the brain slice. I said, let's just go back to the brain slice and try to keep everything as clean as we can, filter the media. And lo and behold, it kept on ticking for 2 days, and then 3 days, and then we started applying more and more signals, and were able to map out a 24-hour, basically dynamic series of states of the clock, where each time of day has its own sensitivities, and there are multiple sensitivities, and they wax and wane just as the sun moves through the sky. And it's interesting to me today, even though we understand the molecular clock works, people really aren't thinking about the dynamic nature of everything else that those cells in the SCN do, from firing to making receptors sensitive to being activated to second messenger pathways being activatable. But none of that has really changed from the early days. So that's how I got into doing this. And I remember the strange thing about the early experiments was that we could only get phase advances. And we never really knew why. We just were trying second messengers that were known, and we found that cyclic AMP worked in the daytime, and our control was the cyclic GMP analog, the other purine cyclic nucleotide, and lo and behold, it was inactive in the day, and it caused phase advances at night. Sort of like a yin-yang relationship, which to any student who looks back at those experiments, which Rebecca did as basically controls, they told her a completely new story. So it was just a superb sort of proof of principle that your controls are important, but when you're working with a dynamic system, you never know what your controls are going to tell you. And the last thing I guess I'll say about those early days was in about, I would say, close to 1990, there was evidence emerging that glutamate might be in the neurotransmitter from the eye, in the projections from the eye to the SCN. So we started applying glutamate as microdrops to the surface of the slice, trying in a very crude way to mimic the effect of a pulse of light. And we got our first phase delays. And I took those data, which were basically a few spots in the daytime, which had no effect on subsequent phasing of this spontaneous activity rhythm, and then a few spots at night where it was delayed, and a few in the late night which caused the next rhythm to be advanced. And I showed this at a SIBA meeting in London, where the presentations were recorded, as were the discussions. And I remember my good friend Fred Turek getting up and saying, "Martha, it's starting to look like you have the beginning of something interesting there. I don't know why people haven't been believing your data for this long." And I remember going, "Haven't been believing our data?" But I always considered that, any criticism that we got was never personally based, but it was based upon the position of the questioner with respect to understanding what's a novel and complex system better. And I always tried to understand our critics in improving the design of our experiments, going into behavior when we found phenomenology in the slice that we interpreted as being the signaling pathway for light, taking any of those agents, either agonists or antagonists, back into the animal. So we acquired the ability to do behavior, and I have to say that allowed us to publish our papers in the best journals. And so you always want to understand your critics and understand where they're coming from and how you understand how they might make your research stronger.
So when you first came into the field, you mentioned some of this already, but what were some of the driving issues in the rhythms field at the time, and how did your work then fit into that, and how did you apply that?
Well, that's an interesting point to remember. I can remember it best because of this Gordon conference that was in Irsee in Germany. It was the first European Gordon conference of all the conferences, and we were sitting at this grand table in the dining hall, and each of the people around the table said, "Well, I'm, for instance, Ray Silver, and I don't know too many of you very well. I'm new to the field." And I said, "No, you're not. I'm new to the field." And then I remember Larry Moran saying, "No, I'm new to the field." And we realized that all of us were immigrants who came in about the same time, but because none of us had really felt that we were embedded in the field, had felt like we were newcomers. And so especially what Ray was doing about that time was establishing the autonomy of the SCN as a timekeeper. So what we had done is take the slice of SCN, the hypothalamus that contains the mammalian circadian clock, put it in minimal salts and glucose in a warm, moist environment, and we asked, can it continue to keep time? And lo and behold, it could, which is part of the evidence that it's an intrinsic clock. What Ray did was experiments lesioning the SCN, showing that rhythms of behavior, running wheel activity, feeding and drinking, endocrine rhythms were gone, were basically abolished, and then taking fetal SCN and poking them back in the brain. And lo and behold, rhythms were restored. Rhythms of locomotor activity, feeding, and drinking were restored. The only set of those rhythms that they measured that wasn't restored were the endocrine rhythms, which we now know or require a specific neural circuit to talk to the hypothalamus, or the pituitary from the hypothalamus. And so it's still, we still ponder why it is that those transplants could restore these general rhythms, basically the rest activity cycle, the motivation to jump on a wheel and run, to feed at a certain time, to drink at a certain time. And presently, those are still interesting questions. What are the signals by which the SCN communicates with the nearby parts of the brain, further parts of the brain, and then what are the signals by which it signals to peripheral clocks? So it was new in the, I'd say that was probably the late 80s, and some of the questions, they raised questions that have still to be resolved.
I think that one of the most important impacts there, things that your work has done, though, is to actually look at inputs into the clock. So could you talk a little bit about all those inputs and how your work fits, or what you've done with that?
Well, the major input, of course, is the input from the retinal ganglion cells that are photosensitive and count photons and send that information directly through their axons to the SCN. And we will probably always best be known for studying that signaling pathway, which is, it's one of the joys of my life that's what I got to work on, because it's so interesting. And the pathways are the same pathway is used in the early night and the late night, and the significance of that is that one causes the clock to be set back to a place it transited 3 to 4 hours earlier, and the other causes the clock to be advanced by about the same amount of time. So you end up about 12 hours apart. So you're basically at diametrically different parts of the clock, even though the same initial signal is there. So one of the interesting things, as we've worked down those pathways, we found bifurcation points. And these bifurcation points are utilizing the very signaling elements that other parts of the brain used for other kinds of long-term state changes, which we call memory formation, learning and memory. And I find it fascinating. It hasn't really been fully explored, but fascinating to think about why the brain should use the same signaling elements to cause long-term state changes, which is a phase shift of rhythms, as it does to acquire the memory that we sat here in Sandestin on a beautiful day in May and had this interview. but it does. And I can't help but think that, first of all, it was used in the clock, because the clock is one of the most ancient of all biological systems. And then later it was adopted by these other brain regions for use there. And we still don't know the answer to that, why it should be. The evidence is emerging that the signaling pathways in the hippocampus, where associative learning is best understood, are now being found to impinge upon the clock genes. So that's one set of signals, and those are dynamic signals. The sensitivity changes over the course of the night, and the result of the signals changes. We know that those are only some of the signals, and one of the best understood signals, at least from our point of view, is in the early night the clock is also sensitive to acetylcholine. And interestingly, it has the exact opposite effect on phase. Instead of delaying the clock, acetylcholine in the early night advances the clock. So here you have a switch where in one state, basically the basal state when glutamate comes in, you delay the clock. But if you apply another neurotransmitter which works through similar second messenger systems, you advance the clock. To me, it's of fundamental interest and importance to understand why the clock can have those two different responses and what gates the ability to go forward or back. We also know that in the daytime, a whole different set of signals, such as PACAP, coming in from the retinohypothalamic tract, serotonin coming in from the raphe, and neuropeptide Y coming in from the intergeniculate leaflet, should cause the clock to be reset. And that's really waiting to be studied again in a more integrative context. And then the last sort of set of signals are those that are neither active in the day or active at night, but the clock is sensitive to them at dusk and dawn, and it turns out those are melatonin. So melatonin has the ability to directly alter timing of the SCN clock at crepuscular periods, where changing night length could cause melatonin to overlap with these intrinsic periods of sensitivity, and we still don't know how that works in an animal on a changed photoperiod, for instance. So that's another fascinating integrative question as far as I'm concerned. So I look at the gating of signaling pathways as something extraordinarily fundamental, important about this neural master pacemaker.
And just before we go on, at this meeting, we've seen people looking at lots of chemical compounds that can impinge on phase, and so it seems to me that one of the next things that may be coming is how the signaling pathways used by these chemical substances. Would you like to comment on any of these?
Absolutely. So it's sort of the high throughput screening of reagents that can alter clock timing, and it's being done in a very effective way, high throughput way, by people like Steve Kay at UC San Diego and Novartis, and John Hoganesh is now at University of Pennsylvania, and there are other groups as well. And from this will come new pharmaceuticals that should be able to correct for individuals who have clocks that run naturally longer or shorter than the normal very small deviation from 24 hours. They may produce reagents that are effective for jet lag, for the loss of coherence in the clock as we age, for sleep disorders that are brought on by shift work or other pharmacological treatments that you're taking, for instance, for high blood pressure, that people don't even think about as affecting the clock right now.
So you mentioned the slice work and that this was a very new and novel approach to studying rhythms. So could you tell us a little more about how that was received, and tell us a little bit about some of your struggles in gaining acceptance of those techniques in the rhythms community?
Well, just like it is in any field, and I learned this by bringing new techniques to bear on this problem. I learned that people were fairly resistant to the notion that you could actually effectively study the clock in the dish. Basically, take what had worked in invertebrate neurobiology, take a slice of brain out, keep it alive, let it continue to live, and then that it would continue to tick, that it would continue to go through these changes in sensitivity to signals coming in. And that's when I realized I had to think about why it was that people, whereas it worked for invertebrate neuroscience, why was it not being, embraced? I thought, wow, what a great technique. You can get rid of all the details, get rid of the legs and the running wheels and just study it there. And then I looked back and I said, circadian rhythms is grounded in animal behavior. All the early work in the 50s was done by the German neuroethologists, Aschoff's school, which was really, completely formative, and this will emerge from other historical interviews. And then people applied other kinds of pressures to different kinds of animal models, insects, photoperiodic animals, plants, unicellular organisms, but it was always an organismic test, even though they knew that within the organism somewhere was a clock. And so the clock was the black box within the organism. And so what we were doing was taking the black box out of the organism and throwing everything else away. And I said, I understand there are other loops, there are other potential regulators there. So if we really want to establish that what we're doing in the reduced slice preparation has importance and validity, what we really need to do is ask if what we find there pertains in the animal. And so one of my students, Todd Weber, had gone to Mike Rea's lab and learned how to do behavior. And so I asked Todd if he would come back and teach it to us. And he did, and we put it in our first study of glutamate, and that was published in Science. And it was basically the story of glutamate and the nitric oxide being a downstream messenger. It was very enthusiastically received, very enthusiastically received. And subsequently, I think we've had behavior in every paper that we've done where we were talking about a new signaling pathway. But a sort of corollary that came along with that was that we not only learned how to do implantations of cannula into the SCN in hamsters, which was really the gold standard for behavior, but we learned how to do it in mice. And I had a superb student, Sabra Abbott, who learned how to put the cannula down within the SCN in the mouse and to implant stimulating electrodes in the brainstem and look at the release of neurotransmitters in the SCN when you stimulate the brainstem in the mouse. To this day, I know that is one of the most technically difficult types of experiments there are, and effectively show that different stimulus parameters could cause release of acetylcholine, glutamate, or acetylcholine plus glutamate, and that those differentially affected the phase of behavior, and that paper should be coming out fairly soon. So it really drove us to try these new technologies, and then just because I guess we're tinkerers, Shelley, when I think about it, we actually took it to a level that I don't think there are very many people doing those kind of experiments in mice even now.
So then, what do you see as the future? Let's start with your work first, and the future of your work and the future of the field.
Okay, the future of my work. Well, since you know me well, I always like to do new things. I've always interested in what can new technologies or new approaches tell us, and I've had the good fortune to have some extremely strong colleagues, collaborative colleagues at Illinois in the physical sciences and engineering who wanted to try new things in biology. And so I'm always willing to listen to someone and see if they have a good idea, new idea. So some of the new things we're doing is applying some of the cutting edge peptidomics and proteomics analyses to the SCN. One of the experiments I'm most excited about is using a new way of using solid phase extraction beads, which are simply what you put in columns to grab things, and applying them to the surface of the slice, sucking the media up, stripping off what's on there, and then looking at all the released peptides. And this sort of discovery approach, there probably was a time where I would have thought it would be all too much information, but the exciting thing is we found peptides intrinsic to the SCN. We found galanin, which comes in from the sleep regions. We found, importantly, a whole new class of peptides, which our subsequent experiments have told us not only are released when you stimulate the optic nerve differentially under select conditions, applying them causes phase resetting, and they're in the SCN, in the central core of the SCN, which makes them potentially part of the relay system. And so I was very interested to see you could do this discovery approach and then carry it in with all the knowledge we have and start adding new players to the cast of characters, the known cast of characters, which is actually pretty small for a highly peptidergic part of the brain like the SCN. So I expect that to take us into new places. And similar kinds of approaches, bridging with the physical sciences and engineers. And I can say today that the National Science Foundation is encouraging neuroscientists to go in this way, to build on their investment in the physical sciences and engineering to further biological research.
So, and then as the current president of SRBR, you've put together a great meeting. So would you like to make some comments on how you see the status of the field right now and where it's going based on the meeting that we're having here today?
It's just been one of the delights of my life to be invested with the responsibility of being the president. Since I was an immigrant and I've never really, I've always considered myself a little bit of a maverick and an immigrant. But right now, circadian rhythms is at an extraordinarily pivotal time. We know not only is the clock in the brain important for patterning the rest activity cycle and the endocrine release from the pituitary to the periphery, but we now know that every cell in the body undoubtedly has a clock. And so the question becomes, why? And how do those clocks get synchronized? And what we're seeing at this meeting is you could almost call it an invasion. It's a migration, a massive migration into the field from individuals interested in studying the bone, muscle, the heart, cardiology, and beginning to look at those peripheral clocks and then to try to understand their significance, largely in a clinical translational context, which is a natural evolution of the field. So with this meeting, the attendance has gone up 24%. I just did the calculation to report at the business meeting. Membership is up 20%. The number of abstracts is up almost 40%. And this just happened as a consequence of us doing what we always do and announcing that the meeting was going to be held. So what it means is People are they're watching, they're interested in what's happening here. They want to come, they want to learn. I predict this field will continue to grow very rapidly in the years to come. So we almost had 600 attendees this time. At our last meeting, we were well under 500.
So how do you see the role of the Society for Research on Biological Rhythms expanding and playing an importance within the field of
Yeah,
rhythms?
I think that's a very important thing to think about. I tried to have the society as a group assume more of a leadership role by engaging people in the field, by having tangible benefits of being part of the group, part of the tribe, developing a communications committee, new website. You developed the newsletter, by having Trainee Day, which was really initiated from the grassroots, but I served on the board of the Sleep Research Society, and it's very effective there to foster the youth, the inheritors of the field, to bring them in. We had 133 trainees and 32 faculty participate in a whole day of professional development. And some other things that I haven't even talked about is the society has assumed a leadership role interfacing with the NIH when there's a call for information. We just developed a white paper on pharmacogenetics. Why, if you think about how genotypes might affect responses to drugs, does rhythms have anything to say about that? And so I wrote to Fred Turek and Joe Takahashi and said, we've got to respond to this. And we got John Hoganish and Joe Owens, one of Fred's students, and we developed a white paper, which then the board modified, and we sent it in to the general medicine to have a position statement. And if it's important that the field do this, that we step up and embrace our position as being relevant to really almost every area of medicine. So I will encourage my successor to do similarly to make sure that our voice is heard at those kind of tables as well.